Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss active transport. In our previous few videos about passive transport, we've come to the conclusion that neither of the two types of passive transport, that being simple diffusion and facilitated diffusion, neither of them require energy input because in both cases, our solutes are going to be moving down their concentration gradients. In other words, from an area of high concentration to an area of low concentration. However, that is not the only kind of transport that can and does occur. In active transport, we are going to be moving solutes against their concentration gradients. In other words, from areas of low concentration to areas of high concentration. However, we're going to see that this is not a favorable process, so to compensate for that, we have to input energy of some kind. This type of transport would be something like an anabolic reaction in which we are going against the flow of things, and in doing so, we have to pay some sort of energy cost. So on the left side of the picture, you can see what we have talked about in the previous few videos, passive transport. Whether or not you need a carrier or a channel or not is going to depend on what kind of passive transport you're talking about. But the main issue is that when solutes move from areas of high concentration to areas of low concentration, there is no sort of energy cost that needs to be paid. But in the right side of the picture, you can see the opposite scenario in which a solute is moving from an area in which it is already not very concentrated to an area where it is already very concentrated. Solutes do not want to move from areas in which they have lots of space to operate in to areas in which they're going to be cramped. So to get them to do this, you have to expend energy of some kind. So if we want to think about this in the way that we have before in terms of uphill and downhill analogies, the actual act of transporting a solute from low to high concentration is very much like trying to push a ball uphill. It's not something that's just going to happen all by itself. You're going to have to put in some energy in order to get that to happen. And that is very different from a passive transport situation in which your solute is already at the top of the hill because it's already at an area of high concentration. So getting it to flow downhill from an area of high concentration to an area of low concentration is just a matter of letting it ease downhill with the flow of gravity, or however you want to think about it. So trying to push a solute from low to high concentration is thermodynamically unfavorable, so we have to expend energy in order to compensate for this. As such, there are two different types of active transport. They are differentiated from each other on the basis of what is the source of energy that we are using to pay the energy cost. But in both cases, the solute in question will be moving from low concentration to high concentration. So in the case of primary active transport, the energy to pay for this cost is derived from ATP hydrolysis, and this is used to pay the potential energy cost for a molecule to move uphill against its concentration gradient. And we have obviously in previous videos belabored the importance of having a cell make plenty of ATP to pay the cost for things like what we're seeing here. The most prominent example of a primary active transporter is the sodium-potassium pump, a very important protein that most cells in the body possess for reasons that you will come to understand very soon in this video. So the first thing we want to make sure that we remember about both sodium and potassium ions is that they are maintained at very different concentrations across the cell membrane. You'll recall from one of the earliest videos in this series that sodium is maintained at high concentrations in the extracellular fluid and low concentrations in the intracellular fluid. On the contrary, potassium is maintained at low concentrations in the extracellular fluid, but high concentrations in the intracellular fluid. So the idea here is that we maintain at all times for any cell very strong and very steep concentration gradients for sodium and for potassium. And in the future, we will see that a variety of different cellular activities actually depend on sodium and potassium being able to move down these concentration gradients by facilitated diffusion. So, if we were to imagine a situation in which we have available and open ion channels for both sodium and potassium, and recall that these ion channels are necessary for either sodium or potassium to move across the membrane because of their strong electrical charge, if we imagine a situation in which we do have these ion channels and they are open, then both sodium and potassium would move down their respective concentration gradients, and as they do so, these concentration gradients would begin to dissipate because sodium would be moving from where it is highly concentrated to where it is more lowly concentrated, and similarly we would see the same thing for potassium. So if we reach equilibrium for both of these ions, in which case the concentration in the intracellular and extracellular fluids would be equal, then we no longer have concentration gradients and we can no longer get facilitated diffusion to occur. And if that's the case, then those same cellular processes I mentioned before that depend on this facilitated diffusion, they can no longer occur. It would be like having a dam that has no water behind it. The whole purpose of a hydroelectric plant at a dam is that you wall off a very large volume of water, and then in getting it to flow downstream, it turns a turbine and generates electricity for you. In the same way here, having these strong concentration gradients for sodium and potassium and then allowing them to flow downstream actually will power a lot of cellular activities, as we will see very soon. But for the time being, the importance is, how do we allow sodium and potassium to move down their concentration gradients without ever reaching equilibrium? And the answer to that quandary is the sodium-potassium pump. This large protein helps to maintain these very strong concentration gradients for both sodium and potassium by pumping them by primary active transport against their concentration gradients. So this sodium-potassium pump is a carrier that, like I said, is going to pump sodium and potassium ions against their concentration gradients to make sure that we always have a high concentration of sodium in the extracellular fluid and a high concentration of potassium in the intracellular fluid. Most cells in the body have this pump, so that underlies its great importance. So in terms of how this pump works, imagine that in a normal situation, we have our respective concentration gradients for both sodium and potassium. Sodium may be low in the intracellular fluid, but there is some sodium there. Similarly, potassium may be low in the extracellular fluid, but there is some potassium there. So the first thing that's going to happen is that 3 intracellular sodium ions will bind to the open portion of the carrier that, to begin with, faces towards the intracellular fluid. So you can see your 3 sodium ions bind there. Next, this channel has a portion that will bind to ATP, again, a very high energy molecule, and then this ATP will be hydrolyzed into the low energy ADP, and this energy will be used to change the shape of the protein so that now the open portion of the carrier faces towards the extracellular fluid. Once this has occurred, these 3 sodium ions can now diffuse into the extracellular fluid, the area where sodium is normally at a higher concentration. So that achieves our half of our goal. We have moved 3 sodium ions from an area of low concentration now to an area of high concentration. So, as it currently stands, the channel is open towards the extracellular fluid. So this is going to allow 2 extracellular potassium ions to bind to the channel now, And now you can probably see where this is going. We need the carrier to flip back so that it's facing its open end towards the intracellular fluid. And this will occur as the channel releases the ADP. This will cause another shape change in the carrier so that it converts to its original ICF-facing orientation, and this will allow the 2 potassium ions to be released into the intracellular fluid. So this is the process of sodium-potassium pumping. We move 3 sodium ions from the intracellular fluid to the extracellular fluid, 2 potassium ions from the extracellular fluid to the intracellular fluid, and we pay the cost for both of those things to happen one right after another by hydrolyzing a molecule of ATP. Like I said, pretty much every cell in the body has one of these pumps, and they are working round the clock to make sure that sodium and potassium maintain their strong concentration gradients across the plasma membrane. And since these pumps are working all the time, that underlies the importance of cells having access to nutrients like glucose and fatty acids and oxygen, so that those cells can synthesize a lot of ATP through cellular respiration, because you've got to have ATP on hand to pay the cost for things like this to occur, things that are happening 24 hours a day. So to wrap this discussion up, take note of the direction each ion moved. Each ion was transported from the area of low concentration to the area of high concentration. Sodium and potassium moved in opposite directions, and that is because their concentration gradients are pointed in opposite directions. Okay, so now let's discuss the other type of active transport, which is called secondary active transport. The idea here is that rather than using the energy from ATP directly to pump one particular solute against their gradient, a separate solute is co-transported from an area of high concentration to an area of low concentration. So another way of putting this is that an active transport process is coupled to a passive transport process. Let's explore that a little bit further. So the first thing we want to acknowledge in this situation is that this molecule here, we'll just think of it, let's say, as glucose. So glucose here, our goal is to transport this glucose from low concentration to high concentration. So in primary active transport, we saw that, well, what we could do is we could move this solute through a carrier that hydrolyzes ATP, and that would solve the issue there. But primary active transport is not all that common outside of the sodium-potassium pump, so we need another way to accomplish this. So obviously we need energy to allow the substrate to the other side, but we need to think, well, where can that energy come from if it's not directly from ATP hydrolysis? So the idea here is somewhat similar to what we saw with the sodium-potassium pump. Our solute is going to bind to the outward-facing part of a carrier, but obviously, like we saw, we need energy to allow for the solute to move to the other side. Well, this is where those gradients that the sodium-potassium pump set up for us can really come in handy for us. In this case, we're going to make use of the sodium gradient. So in this particular case, we see that sodium is at a high concentration in the extracellular fluid. So when a solute like sodium moves down its concentration gradient, this is actually going to release a little bit of potential energy. So as sodium moves through, energy is released, and that energy, which gets funneled very nearby into this carrier, that will allow that protein to shift its shape and its orientation, allowing the solute to move through to the other side. So this is a very interesting sort of situation where the movement of a solute, basically by facilitated diffusion, directly pays the cost for an active transport process. So kind of another way of thinking about this is that the solute moving downhill pays the energy cost for the solute moving uphill. It's not a perfect visualization, but you could almost think of it as this solute, which is at the top of its hill, moves downhill, collides into this solute here, and that transfer of energy is then used to push this solute uphill. Again, that is not a perfect visual analogy because these solutes are not actually colliding with one another, but that is another somewhat useful way of thinking about things. So before we wrap up this video, I want to make sure that I emphasize that one of the big reasons the sodium-potassium pump has to set up sodium and potassium gradients and then maintain them is because those same sodium and potassium gradients are used to fuel secondary active transport processes like these. And in the next video in this series, we're going to actually see a very specific example of intestinal transport and absorption of glucose across the intestinal epithelia. And in looking at that very specific situation, we'll be able to see pretty much every kind of transport that we've talked about so far in action. Okay, that's going to wrap up this video. Here is a list of vocabulary terms you probably ought to be aware of. And then for checking your understanding, number 1, how does the concentration gradient and direction of transport determine whether energy must be expended or not? Number 2, what is the difference between primary and secondary active transport? And third, why would it be correct to say that secondary active transport does indirectly require the expenditure of ATP? All right, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.